All right, well, thank you. So, work in my lab is focused predominantly on breast cancer research, and we do that through a couple of mechanisms. We use some bioinformatic mechanisms, and we combine that then with mouse models. And what I'd like to do today then is tell you a little bit about this and then get at how we're starting to use some CRISPR-mediated technology to start to make knockouts to address that. So what we'd like to start with then is one of the major problems in breast cancer research, and that is heterogeneity. So on the left side, you can see for a handful of breast cancer cases, you can see some of the molecular markers of breast cancer. On the right side, you can see that the heterogeneity that we'd see through immunohistochemistry is also conveyed through gene expression. So each column would be an individual breast cancer sample, and the rows would be the individual genes. And you can see that there's significant heterogeneity there. So that heterogeneity presents a couple of challenges. Number one, how do we model this? And then how do we predict what might be important for mediating metastasis in some of these cancers, which is actually what's going to be causing the death of your patient. So I'd hypothesize then that mouse models of breast cancer are actually going to recapture some of the heterogeneity that we'd see in the human disease. And if that's true, and we can show that, then that's something that we can actually make hypotheses and test for what specific pathways may be important, and we can design actionable strategies as well. So to address that, one of the students in the laboratory has put together a database, and so what we've done is to assemble all of the gene expression information, both from our lab and other labs that have published their work, as well as everything that's been deposited on GEOS. We've taken everything that we can find, everything that's available for gene expression from mouse models of breast cancer, and we've put it together into one central database. And then the question we've asked goes back to that first slide that I showed you about human breast cancer. Is there heterogeneity there? Typically, usually people have said, no, you use one model to study. So a HER2-neu transgenic would be used to study MMTV, so MMTV-neu would be used to study human HER2-positive breast cancer. And what we've shown is that that's not necessarily the case. What we see is that there's an impressive amount of heterogeneity in these various samples. So if you look down at the bottom of the heat map, individual rows are genes, and the individual columns reflect individual samples. And what we can see, yeah, I'm colorblind, it's not going to work. What we can see then is if you look at the top row, you can see MYC in the top. You can see that there are samples that cluster together with each other in the MYC row. But you can see if you look across that some of those MYC samples will cluster more closely with other mouse models of breast cancer than they would MYC itself, suggesting that there's substantial heterogeneity. So that then says that, yeah, okay, we've got heterogeneity in these mouse models. Can we form hypotheses from this? So what we've done is then taken one of those mouse models and compared it to all of the others. And so we asked then, just at a fold change level, the genes that are up in red or down in blue, and what are they in that mouse model compared to all of the others? And so we see those genes for the HER2-neu mouse model system. So this is modeling a type of breast cancer that affects 20 to 30% of women. And so then we asked, well, of those genes that I'm showing in red and blue, what do we have in looking at the proximal location of the transcriptional start site? What do we have in terms of transcription factor binding motifs that might be enriched? And so what we can see in the MMTV-neu model is a significant enrichment for the E2F transcription factors. Now we also happen to have a series of gene signatures for the E2F transcription factors, so we applied that to the data as well. And what we can see in red is a high probability of activation here. So we have a high probability of activation of E2F1 in this HER2-neu mouse model system. So as I said in the very first slide, my lab does both bioinformatics as well as mouse models. So we've combined this now. We've interbred the HER2-neu transgenic mice together with the E2F knockouts. And so we're looking at a tumor curve here. So we're looking at time and percent tumor free. So you can see the curve in blue. This is the standard MMTV-neu transgenic tumor curve. So by 220 days or so, 50% of those HER2-neu transgenics have developed tumors. In red is where we've deleted E2F1. So we have a transgenic with the deletion of E2F1. You can see that whole curve shifted out to the right. What it's saying is that we've delayed the onset of tumors in these mice. More interestingly than this even is that if we look at the tumors that arise in these mice and look for metastases, normally about 85% of the time, the HER2-neu transgenics will develop metastasis. That drops significantly down to about 50% of the time with the deletion of E2F1. So we're doing something with E2F1 to regulate metastasis. To start to explore that, we put the primary tumors from each of these strains out back onto microarray. We went through that, we did a fold change experiment, we did a little bit of a look to see what might be important in human breast cancer metastasis as well. We developed a list of our top hits, and that list, the top 5 are shown here. What I'm showing you here then is these top 5 genes funneled through the human TCGA data. So this is human breast cancer and many other cancer types where all of the omics platforms have been addressed. Each individual column here reflects an individual tumor sample, and this is all HER2 positive tumor samples in this case. So you can see in red for COL1A1, this says that we've got amplification of the COL1A1 gene in those tumor samples. But if you look down, you can see that those same tumor samples also have amplification of a gene called CHAD. What it's saying then is that we have co-amplification of these 2 genes. So coincidentally, they are both located in chromosome 17, as is HER2, and HER2 is amplified and overexpressed in human breast cancer. So we said, okay, well, is this simply just that these genes are co-amplified and they're coming along for the ride? So one of the students went ahead and took the TCGA data, went back and found the array CGH data from that, and said, okay, well, we've got amplification. You can see the spike up around the HER2 locus. And then he looked and it went back to normal, and then it spikes up again for the COL1A1 and CHAD. So what it says is there are 2 separate amplicons, and we get that separate amplification event containing COL1A1 and CHAD in about 25% of these human breast cancer cases. We next wanted to know whether this was actually significant. So to test for that, what we've done is to generate a signature. Because the TCGA data does not have good follow-up yet, we've used a WGCNA method to generate a signature. We validated that in the TCGA data and then applied it to a different data set. And what we can see in red here is that there's significantly more metastasis when that amplicon is present. So to test then for some of those effects of the individual genes, what we did was turn to the CRISPR-mediated technology. So we used CRISPR to generate a knockout for CHAD in the western blot shown on the left, and then we did a scratch assay, just simply asking whether this might affect migration of the cells. So you can see in a wound healing assay where we've blocked proliferation, you can see the scratch at 0 hours. By 24 hours in the wild type control, they've migrated in and filled that wound. With the knockout of CHAD, we can get and see the scratch, but at 24 hours, you can see that that scratch is still slightly open. So we've affected, just by knocking up that one gene, some of the ability of these cells to migrate. We also looked at another gene called FGF13. Some of our CRISPR knockouts are shown here. Again, we see an effect on proliferation and a little bit in the wound healing assay. But then when we actually take this back into the mouse, so now what we're doing is we're taking these cells and putting them in through a tail vein and asking whether they can colonize the lung. What we see is that in our control cell line, so the 419s on the left, looking at a section of the lung, you can see that viable metastases form. When we do that with the knockout of FGF13, you can see that that single gene has blocked the ability of these cells to metastasize. And in the bottom, I can show you the quantitation there, but it's not just metastases to the lung, it's also metastases to the liver, and it's an off-on situation right now. So in summary, what I've said is that breast cancer is heterogeneous, and we need to figure out why it's metastasizing. We can use bioinformatics to make some predictions there, and what we've done is predict that E2F and some of the transcriptional programs that it regulates are important in that process. And now by knocking out some of these genes, we're actually starting to elucidate the mechanism by which that happens. So the students and postdocs and technicians in the lab are shown on the left. We've made heavy use of undergraduates here at State. Collaborators are shown on the right, and funding sources are shown as well. Thank you. Yeah.
Could you walk us through your in vitro CRISPR approach and how you generate knockout cells?
Okay. So we use the older system. We were using a single guide. We need to transition to the dual guide system. So we clone that into the plasmid, just grabbed it off Adgene, put it in, transfect it into the cells. It was the GFP plasmid. We went to the flow sort facility. We sort both on live cells then and on GFP positives. We got the flow sorting facility then to put single cells down into individual wells. We culture those cells up, and then we screen for mutations.
Great, thanks.
It's all done here.
Pardon me?
Campus? So it's done on campus?
Yes, that was all done on campus here.
What do you mean by two guides?
So the dual guide system, the dual Nikkei system.
Dual nicking, okay.
Yeah, so we just use the single guide system. Okay.